Hello class, my name is Sam and I'm one of the TAs for this course. And today I'll be delivering the class on synaptic transmission. So I've attached my email here to the front page, so feel free to write me any questions you have about lecture material covered today, or maybe anything in the textbook about synaptic transmission that you didn't quite understand and you want to clarify. Your TAs are always a point of contact to help you with course material. So again, in regards to this material, please feel free to send me questions particularly. I think it's quite exciting to cover because we're going to learn how neurons communicate and pass information from one neuron to the other. Also making an appearance on the front page here is my dog Millie. I am recording from my apartment, and she does take her job as a protector extremely seriously, so if anybody happens to walk past the window while I'm recording, she may just make an appearance on the audio. So if you do hear any barking, this is the culprit, and I apologize in advance if it disturbs your lesson. Hopefully, we'll be able to catch audio without getting her in here. So let's get started. So neurons rely on functional connections between them to communicate. And you may already be familiar, but this occurs at synapses between two neurons. So you have a signal that is being sent from the upstream presynaptic neuron, and it's sent downstream to the postsynaptic neuron. Now, neurons communicate these signals through two types of synapses labeled here. So we have electrical and chemical synapses. And we'll go through each of these in further detail shortly. I will describe how these synapses provide different mechanisms and structures by which transmission is conducted. But very generally, I'll go over. Electrical synapses allow for direct and passive flow of electrical currents from one neuron to the other. So the channel that the currents pass through are referred to as a connexon, and these connexons serve to connect two communicating neurons at a gap junction. Gap junctions are extremely tiny spaces between adjacent neurons. So we have the presynaptic neuron passing electrical currents through connexons, which pass through the gap junctions, and it will be received downstream or by the postsynaptic neuron. Chemical synapses, on the other hand, have much larger spaces between the pre and post synapse. So they are connected by what are called synaptic clefts. And basically, the synaptic neuron, presynaptic neuron, sends chemical signals or neurotransmitters that are packaged into tiny synaptic vesicles. They'll send it across the synaptic cleft, or i.e. downstream, to the postsynapse, where they are detected by the membrane-bound receptors on the postsynaptic neuron. So let's break both of these down a little bit further, and first we're going to take a closer look at electrical synapses. So electrical synapses are found in areas that require rapid communication. So several tissues including the skin, heart, and smooth muscles, as well as many different brain regions including movement, vision, and olfaction. So think if you need very quick sensory information to be able to produce quick motor output, these are where electrical synapses come in handy, where it's quick reflex almost. So your textbook discusses a few specific examples, such as brainstem neurons and interneurons found in the cortex and thalamus, as well as glial cells. And essentially, these electrical currents facilitate synchronized bursts of activity between adjacent neurons. And this is extremely typical of interneurons, where you have them typically firing in ensembles. So one neuron would cause a neighboring neuron to fire. And let's zoom in on how this transmission occurs. So as we just discussed, electrical synapses provide direct communication. This does not rely on neurotransmitters, and because of this, communication is very rapid. So if you take a look at figure A here on the page, this is from your textbook, and on this slide you'll see how electrical information is transmitted from the presynapse here to the postsynaptic neuron through a gap junction. So this highlighted box area here is the gap junction. B is going to zoom in to the gap junction a little bit closer. So again, this is the extremely small space between the pre and post synapse. And this is where you can take a good look at the connexons, which are the channels that span both the presynaptic membrane and the postsynaptic membrane. And they create a channel which the electrical currents can pass through this gap junction here. Okay, sorry, I'm just going to move this out of the way. It shouldn't be here. There we go. Okay, so these figures are taken from 5.2 of your textbook. Sorry, I'm just trying to make this not pop up here. There we go. Okay. 5.2 of your textbook and illustrate highly magnified electrical synapses that are formed between two neurons in the inferior olive of the mammalian brain. So that's A here. So A is a little bit more zoomed out, and you can see the two neurons with very, very close proximity, and an arrow that points in B is where these neurons communicate with each other at gap junctions. So B is a little bit more zoomed in, and you can really get a feel for how little of a space there is between these neurons, and I feel like this should be turned into a meme to represent the amount of patience we all have left for the year of 2020. But it's really this close proximity that helps facilitate that quick flow of electrical activity. So as we will discuss, and as you are probably more inclined to be familiar with, at the chemical synapse, there's a release of neurotransmitters. So they must travel to the postsynaptic neuron and bind to receptors. And because of this, it creates a greater delay within the synapse. Whereas this doesn't have to occur at electrical synapses because it's a direct passive flow of electrical current. So C here is zoomed in at a gap junction. And again, here you can get a better look for the connexons, which are basically hexamer complexes that span both the pre- and postsynaptic membrane of neighboring neurons. And connexons are created of 6 connexin subunit proteins. So these proteins are formed together, and this is what permits the channel for electrical transmission to be conducted through. So these channels are much larger than voltage-gated pores, and this allows for the passage of more medium-sized molecules such as ATP and second messengers. So this further permits the activation of similar transduction pathways in both neurons. So just remember the idea that an action potential in one neuron can pass electrical signals to neighboring neurons, causing it to fire as well, and this occurs rapidly. And just sort of highlighting the rapid nature of electrical transmission, this is figure 5.3 from your textbook. And A refers to a crayfish gap junction. So taking a look at the graph, we have the postsynaptic neuron on the bottom and the presynaptic neuron on top. And as you can see, the postsynaptic neuron becomes depolarized within the fraction of a millisecond when you compare it to the depolarization of the presynaptic neuron. And this is the very quick reflexive-like responding that allows for the fast escape from predators that the crayfish may encounter. So in part B, we have a graph representing electrical synchronization between hippocampal interneurons. So on the bottom graph here, you have red asterisks, which are representing each time neuron 2, which is at the bottom here, fires at the same time as neuron 1. So while some electrical synapses only permit unidirectional flow, the majority provide bidirectional flow, and this allows for, again, electrical synchronization between the two neurons. Okay, my apologies. I just need to connect my charger for my laptop, which fell out, so I can continue this recording. Okay, let's go to the next slide. Now we're going to turn our focus to chemical synapses. So how do neurons communicate via chemical signals? Am I on the right? Yep. Okay, so these figures here are from your textbook, and they both depict a chemical synapse. C is a little more zoomed out, so you can see the presynaptic neuron. It's full of neurotransmitters that are packaged into these little vesicles, and then you have the postsynaptic neuron. And basically these vesicles fuse to the presynaptic membrane, and through a process of exocytosis, they're going to release neurotransmitters into the synaptic cleft. So looking at D, we have a little bit more zoomed in on the area of the synaptic cleft, and here you can see these neurotransmitters then bind to receptors on the postsynaptic membrane, which could either be ionotropic or metabotropic receptors, and we will cover these different classes of receptors in future slides. But just for now, ionotropic receptors are ion channels that open and close when a neurotransmitter or a specific ligand is bound to the receptor. Metabotropic receptors, on the other hand, are coupled to G proteins. So when they're activated, they can modulate ion channels through metabolic processes. So again, we'll cover these better in a few slides earlier, but either way, whether the receptor, doesn't matter which type of receptor it is, it's going to change the membrane potential of the postsynaptic neuron and thereby changing the membrane potential. Binding to oh, sorry, I already covered that. Yep, that's all I need to get to with this slide. So these pictures here from your textbook highlight certain structures at the chemical synapse to take note of. So A, you have the pink, which represents the presynapse, and the green, which is the postsynaptic neuron. So you have the synaptic cleft, which is labeled in both images here and around the presynaptic membrane. The darkness would represent the area of the active zone. So the active zone is where synaptic vesicles bind to the presynaptic membrane and they release neurotransmitters through exocytosis. On the postsynaptic membrane, you can see this very dark region here, is the area of the postsynaptic density. And this is very important for postsynaptic signaling. And it's essentially several dense projections that are formed together, and hence the name. B, you can get another look at these structures in a 3D recreation of a chemical synapse. So with the dark blue area of the postsynaptic density being represented right here, the postsynaptic cleft right here, and then just on the other side of the presynaptic membrane, you have the active zone, where again it is the area where synaptic vesicles are going to dock and allow neurotransmitters to be released from the presynaptic membrane through a process of exocytosis. And just before we get into detailed steps of the chemical synapse process, let's just go over some details about two major families of neurotransmitters that are used for chemical communication. And that is small molecule neurotransmitters as well as neuropeptides. So small molecule neurotransmitters include acetylcholine, dopamine, and glutamate, as well as several others. And these molecules typically modulate rapid synaptic actions. So in the case of acetylcholine, it is known that it's released in neuromuscular junctions that require responsive and very rapid communication. And then we have neuropeptides, one of which is discussed in your textbook is enkephalin. And on this slide here, we're going to differentiate how these families of neurotransmitters are differentially synthesized and packaged. And just to be clear before we start, multiple neurotransmitters can be released from the same synapse, into the same synapse. So it's really the cumulative outcome of multiple neurotransmitters that produces the effect on the postsynaptic neuron. So for example, one neurotransmitter that is released could be excitatory, and hence it would excite the postsynaptic neuron, whereas another neurotransmitter may be inhibitory. So it's really this ratio of excitatory versus inhibitory signals that the postsynaptic neuron is picking up that ultimately determines whether it fires or depolarizes, becomes depolarized. Okay, so now let's get into the main point of the slide again, which is neurotransmitter packaging and transport, which is different depending on whether it is these small molecule neurotransmitters or these neuropeptide neurotransmitters. Okay, so small molecule neurotransmitters, the packaging and transport occurs locally within the presynaptic terminal. So the enzymes that are necessary to synthesize the neurotransmitters are produced in the cell body. And then those enzymes are sent down the axon by slow transport along microtubules. These figures are actually taken from an old version of your textbook, so you won't find them in yours if you have the new edition, but I think it's a great diagram to follow along as you read through the material of the current 6th edition text. So we have, again, enzymes produced in the cell body that are transported to the presynaptic terminal. And that's where they await precursors that are taken into the terminal actually by transporters along the presynaptic membrane. So the precursors are taken into the terminal where they meet with the enzymes. So you can see in this diagram here, the precursor molecule interacts with the appropriate enzyme to create the neurotransmitter. And then this neurotransmitter is packaged into small clear core vesicles. So that is a term to highlight here. As you'll see, neuropeptides are packaged into large, dense core vesicles. And as the name suggests, they're called clear core vesicles because they appear transparent, and they're relatively small packages that range from 40 to 60 nanometers in diameter. So neuropeptides undergo different packaging and transport. The enzymes and precursors are actually packaged together in the cell body into large, dense core vesicles. So as the name applies, these are larger vesicles that range from 90 to 250 nanometers in diameter and are electron dense. These vesicles then travel to the presynaptic terminal via fast axonal transport. And some of the synthesis and modification occurs en route, but it's typically the final stage of synthesis that occurs in the terminal. And again, more of this will be covered in chapter 6. But this is the basic difference of how small molecules and neuropeptides are transported and delivered to the terminal.